So while people are still joining, I'll just start. Welcome everyone to this Friday's SEM seminar. Of course, the SEM is organized by the LABEX CEMEB, and we're very thankful to Agreenium for providing us with the Zoom webinar license. As always, you can ask your questions already during the webinar. If you look at your screen, at the bottom of your Zoom screen, you'll have a Q&A tab where you can enter your questions and you can vote for other people's questions. You can use the chat maybe for more technical issues than for questions. And I don't have anything more to say, so Sylvain, it's up to you.
Okay, thank you. So it's a great pleasure to have Joshua Weitz today. He's going to tell us about the dynamics of viruses, and he's a professor of biological sciences in Georgia Tech in Atlanta. And originally he did a PhD in physics in MIT in 2003. And after a postdoc in Princeton, he joined Georgia Tech. He's currently the director also of the new interdisciplinary PhD in quantitative biosciences at Georgia Tech. So I think he might briefly mention that. He's interested in a whole range of subjects, but mostly in disease dynamics and quantitative viral ecology. And he's also the author of a great book, so I'm going to do some publicity, about quantitative viral ecology. So it's a really nice book, so I recommend it. Because it goes from the description of the virus life cycles with all the details of the different traits of the viruses, and going even sometimes within the host cells, from the individual cell level to the microscopic levels, where he's addressing big questions about the ecology of virus and their role in the functioning of marine viruses, for example. So he might also mention that. So he's also involved in disease dynamics, as I said, but for example, in COVID-19, he's also developing models trying to help manage the current pandemic. So I think I leave it to you, Joshua, to tell us about your work.
Thank you so much, Sylvain, for the introduction, also for the invitation to join. Hello, everyone. I realize that in these webinar modes, I can't be there in person with you. I actually was meant to be there in person. I plan to spend next year in France. I was supposed to be there this year. It's been postponed, based in Paris, but look forward to visiting, hopefully another opportunity to come to the south, to Montpellier. And Sylvain, I hope you can hear me. If that's the case, just give me a thumbs up. And beautiful. Okay, so let me share my screen and I can get started. And if you can just make sure to let me know, can you see my screen? That would help me know that I can go ahead.
Yes.
Okay, great. So you were right, I was going to do a brief plug for this program. We have a new PhD program in quantitative biosciences. So if there are undergraduates looking to explore PhD work at the interface, really between the biological sciences, physical, mathematical sciences, we welcome them. Our interest is moving across scales. So we have folks working on molecular and cellular systems, organismal behavior and physiology, up to ecology and evolution, and even in some cases, Earth systems. And really the intent here is to unite a multi-scale study of living systems and recognizing that students can come from very different backgrounds, and we have a mix of sort of theory, computation, and experimental work involved. And the website is qbios.gatech.edu. So if you want to learn more, more than happy to talk to you about it. And today I'll talk about the entangled fates of viruses and the microbial hosts. I'll try to give you a broad view of what we do and trying to hit on a number of themes, but really focusing on this theme of entanglement and hopefully give some new ideas or new vistas on old problems. And Sylvain has worked on some of these. I'll allude to some of his work as well. But again, I'll try to give you a broad view of what we're up to, and I won't be able to cover all the application side, the ways in which the concepts that I'll explore today will also influence the ways that I think we might think about phage therapy, about understanding viruses and marine systems. And yes, some of that's in the book, but thankfully a lot of it is not in the book. There's still a lot left to do, not just obviously for my group, but really for the community. We need more folks thinking more carefully about viruses and microbes. And part of that is when we talk about viruses, this is usually what we talk about. Viruses that infect us, whether it's Ebola or Zika or influenza, and in other years we might have been talking about these viruses, but as you all know, most of the time when we talk about viruses these days, we just talk about this, SARS-CoV-2. And yes, I have been working on problems of that nature. I've included here some results, some positive news at Georgia Tech, where we had a reintroduction of students, nearly 10,000 in mid-August. We saw a slight upsurge in cases, but we've implemented a large-scale viral testing program, asymptomatic testing program. As you can see, it's almost been a full month since we've had incidence, above 1%. In other areas, we've worked on testing for antibodies. We have a website that we've developed that gives the risks associated with events. It went viral in a different sense in the United States in July. There have been over 2 million visitors to the site. We are thinking about additional European country expansions. We've added a UK, Switzerland, and Italy site, and we are thinking about France. So if anyone's interested in talking to me about that, we'd be more than happy to discuss. But that's not what I'm gonna talk about today. I'll try to focus on viral infections across the diversity of life. And yes, viruses can infect humans, mammals, birds, insects, plants. They also can infect microbes. That is to say, microbes get sick too. And these infections can be of amoeba, archaea, and bacteria. And for the most part, I'll be focusing on these bacteriophage, bacterial viral infections of bacterial organisms. And when we think about the consequences of infections at the level of single-cell microbes, it's often through the lens of lysis. And the consequences not only at the cell level, but at the population level. And I've given here 3 representative examples, all from marine systems. We tend to do a lot with our collaborators in marine systems. And these are 3 different examples of infections. And in each case, what I hope you can see here is that we have dynamics of the cell alone, and then the cell dynamics at the population level with viruses, and the increase then of virus particles over time. And in this case, this is actually giving you some idea via host DNA and intracellular phage DNA and extracellular phage DNA, and that's for cyanophage infections of ubiquitous Prochlorococcus in marine systems. All of these reveal that viruses can have significant deleterious effects, dropping down host density, leading to the rise and increase of viruses. But of course, these virus microbe pairs exist in nature. It's not as if, even if you can add viruses in a flask and drop down cell densities by tenfold or even eliminate them entirely locally, that is not actually what's happening in natural systems. In natural systems, of course, there are many other players. Viral infections of organisms may kill a subset of cells, and that lysis of cells, in fact, in some ways keeps organic matter small. And this idea was formalized in the late 1990s by Jed Furman, Curtis Suttle, Steve Wilhelm, and others as a viral shunt, meaning the lysis of whether it's heterotrophs or autotrophs redirects carbon and nutrients back into the dissolved organic and particular organic matter pools, which can then be taken up again by the same microbes rather than being moved up the food chain to grazers and higher trophic levels. And so we can think then of viruses not only as the agents of mortality, I think we all know their potential role in gene flow, but also as organisms and parasites that can modulate ecosystem processes. And that's really what we've been trying to do over my faculty term here at Georgia Tech, which is the group's focus is thinking about how virus-host interactions modify the fate of cells at the time scale, right, of the cellular time scale, division times, But the death of a cell doesn't necessarily lead to the death of a population. We can see that nonlinear feedbacks can lead to oscillations between viruses and hosts, similar to Locke-Volterra-like dynamics, and I'll explain that more. And then when we magnify and scale this up to ecosystem scales, then the consequences of these repeated interactions and lytic events can influence not just community composition or population abundance, but even ecosystem processes. And part of the reason for that, and this is work that we did as part of a Nimbus working group, is to re-evaluate the abundances of virus particles in ocean waters, both at surfaces, where you can see here in green, and at depth below 100 meters in the water column. And the point I want to take away here is simply that there are a lot of viruses. 10 to the 7th per mil, in other words, 10 billion in a liter, are typical in seawater. The range goes from anywhere from 1 to 100 to 1. And so it's not just a simple ratio, as you may have heard of, like a 10 to 1 ratio. That's not the case. There's significant variation. Variation is typical, but nonetheless, there are an enormous number of viruses. And for the most part, these viruses infect ocean microbes, right? You can go into the water, swallow some seawater, and you're not going to get sick. They're not going to infect human cells. So I'll try now to use this kind of foundational material and ask the question, do viruses and microbes, whether in the oceans and elsewhere, do more than kill or prepare to kill? Should we think about other ways that viruses can influence microbes, populations, and communities? And of course, the answer is yes, but I want to explain a bit why I think it requires some elaboration. And the premise is built on the following simple question, which if we're in person, we could maybe talk whether over coffee or wine or beers at some extended hour, but I'll just try to frame it with the question, what is a virus? And I've given 3 examples, one of which is this virion. You can think of this as its charismatic form. I showed you that picture of SARS-CoV-2, and it's often what we show when we show viruses, that we ascribe implicitly the word virus to the virus particle. But there are also things like Lysogeny, in which in the case of bacteriophage, the genome of the phage can be integrated into the chromosome of the bacteria and then potentially pass on vertically, and also lytically infected cells, which is essentially these virions now inside a cell. So they're intracellular rather than extracellular. And I will make the claim today that I think we should be thinking about all of these as the virus. Not necessarily just the virion or the Lysogeny, lytically infected cell, what is often called a virocell, but rather the entire viral life cycle. And in some fundamental sense, the viral life cycle is what is under selection. And I believe that thinking in this way actually brings new challenges for theory, experiments, and fieldwork, and helps us rethink some of the conflicting and often confusing evidence we see from natural systems. So I'll try to elaborate on this one question in 3 parts. First of all, by going through some foundational ideas of lytic models of phage bacteria dynamics. And frankly, the book that Sylvain noted before really focuses largely on lysis and not as much on lysogeny. And so thankfully for me, for others, right, there's a lot more to do. And I'll begin that process in part 2 by talking about some confusing observations from marine systems, which have gotten me and folks in my group and collaborators more excited about revisiting this problem of lysis versus latency, not just in experimental model systems, but really in natural systems. And finally, I'll share some of our perspectives on ways that we think we've been trying to advance some unification of viral fitness and understanding of it along a continuum from lysis to latency. Okay, and I'll start, and again, it's hard to take questions in these webinars, and I know that there's a format here that we're going to wait to the end, but if there's an urgent question that pops up and needs clarification, I assume Sylvain or some of the organizers will interrupt me and let me know. Okay, so just to make sure everyone's on the same page, let me just explain the life of a bacterial virus or phage, in which you have this diffusing 50 to 100 nanometer sized virion. It can be larger, can be smaller, comes in contact with the host cell, injects its genetic material into the host, which then redirects host machinery to produce viral proteins, which self-assemble, and eventually as the viral genome is replicated, there's a packing into these capsids, which through a time process of usually a holin disrupting the membrane and then the lysine disrupting the cell wall leads to the virion particle release, and the cycle begins anew. And this obviously is the death of this particular host cell, but not necessarily the death of the population. And in fact, going back to Bruce Levin's seminal work in 1977, it has long been held that essentially in this field, we should think of virus-microbe dynamics as predator-prey-like dynamics. And here's a section from Bruce's work describing one resource, one prey, one predator, as essentially a system of nonlinear coupled populations where phage, acting like a predator, reduce the density of the bacteria, and as bacteria are reduced, that influences phage dynamics, and so on. And this goes back even further to Alan Campbell's idea from 1961, in which phage were termed the simple predator. And the premise of this work and the conditions for existence of bacteriophage along with a host is that if a virulent phage and a susceptible bacterium are mixed in an open growth system, such as a chemostat ... and the presumption of the ... is that under certain conditions they could coexist. The elimination of the prey does not necessarily lead to the elimination of the prey population, right? And that there can be coexistence often in these oscillatory modes. So these predator-prey models have been the basis for quite a long time for studies of virus-microbe population dynamics. And part of that is it's easy to detect lysis, right? It's an easy way to identify a virus. Often the isolation methods involve looking for plaques or other clearings. So we're using lysis as an evidence, not only of interaction, but of presence and isolation. And so here, what I'll try to do, and I'm gonna see if this piece can work. I'm gonna try to use the laser pointer here as a way to demonstrate. We have a simple chemostat-like model where there's a resource layer. You could think of this as a carbon source, bacteria, which is N, and virus particles, which are V. This is a chemostat model, so there's going to be influx of resources at a rate omega, and then resources, bacteria, and viruses will all flow out of the media at a rate omega as well. Carbon, this resource is consumed by cells, which then divide. Viruses infect, meaning move out of the virus particle category, and also, compartment, and cells are then lysed, and this is a model of instantaneous lysis, so there's a release of beta new virus particles. Right? So this tripartite layer of resources, hosts, and viruses, we can look at in terms of this phase plane where I ignore for the moment the resource layer and ask what would happen if we add viruses to an otherwise resource-limited chemostat. What you find is that viruses start to increase. We have a large number of viruses which then drive down the bacterial population. And because there are fewer bacteria, then the viruses decrease. Because there are fewer viruses, aka these predators, the bacteria increase in abundance and the cycle repeats. And in this particular example, we get these counterclockwise cycles that converge to an equilibrium. If I had used a delayed version of lysis, or even an explicit representation of the infected cell state, you can go from these convergence to a fixed point to true stable limit cycles. So the prediction here, once we make this analogy of predator-prey dynamics, is we should expect lockable Terra-like dynamics for the same reason that one expects those kind of cycles endogenously in a predator-prey system. And it turns out these have been measured and observed in experimental systems. This is work by Brendan Bohannon and Rich Lenski. You may know both. You may know Rich from his work on long-term evolution experiments with Ecologie, but prior to that, he had worked with Bruce and many others on these phage bacteria systems. And as you can see here, there are large-scale oscillations in which viral peaks seem to fall after the host peaks. And also notice that this is multiple orders of magnitude of oscillations. So then as bacteria peak and are eliminated, there are very few bacteria. Viruses will start to be removed via the dilution due to the chemostat, and only then again begin to increase once the bacterial population increases and the cycle repeats. So this is an example of what happens in 200 hours. What's interesting, of course, is that they continue to do this experiment past 200 hours. And what they found was that these predator-prey-like cycles gave way to something else, what are called cryptic dynamics. And it's called cryptic because if you had seen a host signal like this and a virus signal like this, you may not expect that they interact. But these are the only players in this particular chemostat. And as you can see, there are periods in which viral densities are increasing. Clearly, they're infecting, replicating, and lysing something in that environment. And what Bohannon and Lenski realized, in fact, that this host population was not a single population, but there had been an evolutionary event, and they recognized there seemed to be both resistant and susceptible types. They later isolated those, reintroduced them so that they could differentiate the abundance of both. And in doing so, what they found is that the resistant hosts could in fact rise to high densities, and that these oscillations represented oscillations between viruses and the susceptible host. But if you'll notice, they have the structure of Locke-Volterra dynamics, meaning that it has the right phase shift, but the susceptible host densities are significantly lower than the resistant host, which means that this is essentially in the background of the total signal, giving way to this cryptic-like dynamics. And there's also a cost to resistance in this case, which means it doesn't supplant the susceptible host that is vulnerable to viral infection. So to summarize this part, just want to kind of give everyone a sense of some of the context before we move into thinking about latency, which is that in the absence of evolution, if we think about predator-prey-like dynamics for viruses and hosts, we should expect these counterclockwise cycles, these cases in which the prey peaks first, followed by the predator, and so on. When there's predator-prey evolution, then we can get cryptic dynamics or anti-phase-like dynamics, and this has been reviewed. It's often the case that many of characteristic predator-prey time series don't have the typical quarter period lag, but actually have things that indicate that evolution, whether of predator-prey, is important. And just as a side note, if both predator and prey evolve, there can actually be coevolution, which leads to a reversal and clockwise dynamics. In other words, things that appear like the prey eats the predator. It does not, but it's the change in life history traits between co-evolving predator and prey that can lead to total ecological dynamics that appear to be the inverse of that expected under a Locke-Volterra-like assumption. So the takeaway here is that population dynamic patterns have been used to infer putative mechanisms. There's a connection between the extent to which we understand those mechanisms and the resulting population pattern, but can we take these same ideas to non-lytic interactions? And that's what I'll try to do in the second part, talking about lysis, lysogeny, and virus-microbe ratios. Okay, so to revisit again this life cycle, I described the lytic pathway. Now I want to describe the lysogenic pathway, because there are other kinds of bacteriophage called temperate phage that can both integrate or kill a bacterial host. And you can see this here in the schematic where there's the injection of genetic material. And then the question is, a decision arises between initiation of lysis and that of integration, in which the phage is integrated, the phage genome is integrated into the host, and we refer to it as a prophage. And this genetic switch has been studied. There are wonderful books, Potassium is just one. And these integrated phage then can be passed on, inherited vertically from one mother cell to daughter cells. And then later, depending whether by chance or by a stressor, there can be a process of induction in which the prophage is excised from the genome, reinitiating the lytic pathway. And this has been studied going back to Luria and Delbruck and their work on the origins of mutation. Also, André Loif at the Institut Pasteur as a beautiful and still relevant, in my view, review from 1953. There's still wonderful things to learn in it, who also essentially took out these same ideas of thinking about ways in which you have an intersection of life cycles. Right? Initiation of lysogeny can then lead later, whether through chance or through some stressor, to the reinitiation of lysis. And there's other kind of factors, loss of prophage, and there's a whole bunch of ways in which this becomes an interesting problem. The study of the basis, not only for these life cycles and the molecular mechanisms, but the impact on populations, has been reinvigorated in part by single cell and single phage studies. Edel Golding's work in particular is quite elegant. And often in these cases, what they do are label the phage so that you can see capsids with this YFP here looking green, and also expressing on the phage genome a reporter that will report red fluorescence when the phage is integrated as a prophage. So you can see here the transition between an infecting phage on a surface. Some cells are going to produce new phage and eventually lyse, releasing new viral particles, and others that are expressing red fluorescence divide. And what we see is the normal growth of the E. coli, leading to division and also passing on heritably this prophage. What's interesting, of course, is that these features of whether or not to integrate or to kill are not just fixed features. And this is something that I can't tell you that we've come to a total solution of. We have some ideas, and I know Sylvain has similar ideas. I think there's still a lot to think and to study here, particularly because these are plastic traits. We can think of these as not being fixed probabilities that somehow travel with the genome of the virus, but they're contingent on the host, but also on the environmental context, whether or not the host is singly infected or multiply infected. And going back to work by Philippe Kurilsky in the 1970s, it's been well understood that as you increase the multiplicity of infection, the number of phage that co-infect a particular cell, the chance of lysogeny goes up. So in other words, it's a less virulent outcome, even as there are more phage inside a particular cell. So if you ask you the question, do you want to have a larger viral dose for a particular kind of infection, you probably prefer a smaller dose, thinking that that would lead to a less virulent outcome. Here, it's the opposite. And what they found is that this means that at the level of a single cell, right, the chance of having a lysogeny being integrated really is a chance. It is a stochastic system, but it is driven in part by this cellular multiplicity of infection. And just to point out, these are evolvable traits. They can be changed, and they do change, and even the fraction induced is also going to be, in some sense, a plastic trait. It depends. As you have more stress, there are more that are induced, but baseline levels themselves can evolve. Okay, so now that we understand a little bit of how to be temperate, meaning that there's some genetic switch which leads to the initiation of lysis or lysogeny, Frank Stewart and Bruce Levin asked a wonderful question that I think still deserves more time. The question is why? Why be temperate? And to do so, they asked this question in a mathematical way by, again, envisioning a chemostat in which there weren't just cells and virus particles, but also lysogens. And the question then became, when would a strategy which initiated lysogeny somehow do better than the one in which strictly killed the cell, produce more viruses or virus particles? And their premise was one of a feast or famine hypothesis, that they exogenously drove good times and bad times and claimed that temperate phage do better when few hosts are available and extracellular mortality rates are high. And the idea there is essentially that if there were very few hosts available and the phage tended to kill those few hosts, there wouldn't be any hosts left. That is a bit of a group selection argument. And in fact, even if it's right at some level, the caveat that they explained in their paper, in spite of this intuitive appeal, this low density hypothesis, we're unable to find solutions consistent with it using the model presented here. And that will be some way that I will begin to push in this third part. Okay, so this notion that viruses tend not to kill cells when they're low abundance is backed up not just in models or in this phage lambda system, which also essentially does the same thing, right? Where I just showed you data showing that the probability of integration went up when there in some sense were fewer cells around because viruses were reinfecting the same cell. You can also see this in marine systems where people have looked and called this seasonal time bombs. The idea is that lysogeny is prevalent given essentially low productivity, low density environments, and lysis is elevated, high productivity, high density environments. What you can see here is that by looking at the percent of bacteria that are lysogens, in order to do that, you have to induce them with a stressor, in this case, mitomycin C. It's much higher in these low productive, low density environments over the spring. And here in the high productive environments, there's almost no lysogens, but we see, this was work by Jennifer Brum, they saw high fractions of infected cells, which they describe as lytic infections. So there's a transition here between, in some sense, not killing cells when it may not be so many cells around, and killing them when cells are abundant and growing fast. And this has been the paradigm, both from phage lambda and from work in ocean systems, going back to John Paul's work with Lauren McDaniel in the early 2000s, that there's this switch, high density, high abundance, more lysis, low density, low abundance, more lysogeny. In 2016, this was proposed to be turned on its head by a paper from Forrest Rauer's group, focusing on an alternative hypothesis, quite literally the inverse of everything I've just described, and it's termed Piggyback-the-Winner. The premise is that lysogeny is positively correlated with increases in host density and productivity, meaning as these go up, one should expect more lysogeny and less lytic activity. And they developed this model in part to try to explain a pattern. And the pattern was that the ratio of viruses to hosts, or the relationship between viruses and hosts, was not linear. In other words, as microbes increase, one did not find a linear increase. This is a log-log scale, but rather a sublinear increase, or power law. I know there's a lot of scatter here, but nonetheless, I'll call this a power law, which is sublinear. In other words, the ratio of viruses to hosts seemed to decrease as hosts went up. So their premise was that if it decreased, this implies somehow that lysis is less relevant. We have also looked very carefully at these ratios and the relationship to virus and prokaryotes and natural systems using this revisiting of a large number of independent marine surface ocean and deep ocean studies, and found a similar result from a pattern perspective. This is the 10-to-1 ratio. The overall data set is fit by a power law, and in fact, individual data sets tend to be fit by power laws that are also have exponents less than 1, implying that the bulk of the data implies some sort of sublinear relationship. The ratio of viruses to microbes decrease as microbes density increases. However, in order to make the connection between this idea and those patterns, they came up with a model which did not include lysogeny. In fact, it's a lytic model in which you have microbial cells increase because of growth, lysis, there's mortality and decay of both. If you'll notice here, and sorry for the P, which should be a V, that the viral release is proportional to the contact between cells and viruses, but actually it increases with N. So it doesn't decrease, unlike the premise. The difference here is that the end result, the equilibrium, rather than the activity, does seem to have this sublinear feature. But many other such models can produce that kind of pattern. Whether you use a classic Locke-Volterra-like model without invoking any form of lysogeny, just some form of life history parameter variation, or use this Piggyback-the-Winner model, when you look at the ratio of viruses to host versus host, you can easily find this decreasing relationship. So our point here is that this pattern of a decreasing ratio with microbial densities is insufficient to discriminate between mechanistic hypotheses, which we describe in this brief report. There are also some apparent metagenomic evidence, direct evidence, that the fraction of indicators for being temperate, things like integrases, excisionases, and other provirus-like reads in the virome, in other words, in the viral particles that are not inside cells, was going up, as claimed by Knowles et al., with microbial cell density. We revisited this data and were not able to recapitulate the same relationships. In fact, as you see here, this is the percent of provirus-like reads versus microbial cells. Here are integrases, and here are excisionases. And in every case, we see no significant relationship. And even to the extent to which you were to remove a few outliers, the explanatory value is nearly 0. And so there's an ongoing correspondence there, but the takeaway point is essentially that we found at least no evidence at this direct level or in the metagenomic side for Piggyback-the-Winner. However, this notion that virus to microbe ratios might go down with microbial abundance inspired others to take a closer look. And here's an independent study, again, looking at metagenomic data that found a decrease in virus microbe ratio as a function of increasing microbial abundance. And as they pointed out, this seemed to corroborate the recently proposed PTW, Piggyback-the-Winner theory. You'll notice here that these lines all seem to be in parallel. And when I first saw this, obviously we had been thinking about this topic, I was a little worried because, as you can see and hear a replot by our team, they seem to be parallel to a -1 slope. The ratio here is viruses to microbes, and the x-axis is microbes. So in other words, if y over x looks like x to the -1, this doesn't necessarily imply some sort of sublinear relationship. It implies that virus abundances are unrelated to host abundances, which is a counter indicator for Piggyback-the-Winner. Okay, so the takeaway here, there's an absence of evidence for these relationships. We aren't necessarily able at this point to infer a mechanism just from these patterns alone, but I will point out that something important has been made here, which we also don't see particular evidence for the seasonal time-bound hypothesis in some of these new data sets. So it really raises some important questions. And those questions I'll try to address in this last part. The question I want to ask in this last section is simple, and I think it has been the important part that's come out of these debates in the last few years about Piggyback-the-Winner and lysis and lysogeny and virus-to-microbe ratios and how much can we learn. And the question that I like to ask is what environmental conditions should favor lysogeny rather than lysis? And I don't think we quite know the answer yet, but I think that there's a lesson here from the past that may be worth guiding us in the present. You may have heard the expression, a bird in the hand is worth two in the bush. Someone can tell me if there's an equivalent expression in French. The idea is that you have something, you might give it up for something else that's uncertain, but because it's uncertain, you kind of want more of whatever it is you have. In this case, 2 times more. And so I'll pose a puzzle here at the end that a virus in the cell is worth n in the bloom, meaning the virus is inside a cell. It has reached something that it can potentially replicate on. It could lyse it, but then there should be something else out there to infect. And the question really is, what is n? Right? What is the appropriate number of opportunities for a virus to kill a cell that it has found in a heterogeneous, changing environment, that it would kill it in order to find new cells that may be uncertain? To do this, I'm going to go back to this viral proliferation and make a claim that we should think more about these life cycles, and I'll try to do so in the following way. This again are these obligolytic viruses. There's entry, and here a virus is injected genetic material in the host, and I'll call this the mother virus. It lyses and produces many virions, and this could be dozens, if not hundreds, and in some cases for obviously eukaryotic viruses, things like EHUX and others, it could even be thousands. If we think about this of infecting a typical bacteriophage in bacteria, we might think of this as 50 to 100, but that is not the fitness nor the end of the life cycle, because we started inside a cell. Most of these virus particles will never come in contact and end up successfully infecting a new cell. In this example, I've shown 3 that did. These daughter viruses we can think of as being the fitness at the individual level, right? That this one virus has led to 3 new viruses, if we think of these in terms of infected cells. We can do the same thing for latent viruses, thinking about temperate phage. And here, although this lytic pathway is an option, I'll now follow just the prophage pathway, in which there's an integration event. Again, we'll call this the mother virus, but now it's a lysogen. It divides 3 times and then dies. And you can see that now there are 3 daughter cells. The fitness of each is their own. And we can use the term R verse 3, meaning there's 2 distinct ways of getting the same number of infected cells from this original infected cells. And I've put fitness in quotes here, recognizing that we really have to think about time and Malthusian fitness to talk about fitness at the population scale. But I'll try to make this bridge by asking the question, how does this depend on cell densities? To do that, we can take these frameworks and translate them into a population model where, again, we have susceptible cells, infected cells, and viruses. Each can either die or decay. There's infection leading to new infected cells, and lysis over a period of time, 1 over eta is the latent period on average, leads to the release of beta new viruses. Right? And so in this case, we can ask the question, if there's a susceptible population, what are the conditions by which viruses could proliferate, exclusively given horizontal transmission? And to do that, when we calculate the invasion criteria, we find there's a critical number, which now, for those in this talk who are actual epidemiologists or those who are just armchair epidemiologists, you'll recognize as the basic reproductive number. I'm using this term HOR for horizontal. And it turns out to be the product of 3 things. Starting inside a cell, this is the ratio of the rate of lysis divided by lysis rate plus death rate. And this really means the probability that the cell will lyse before it decays for other means, multiplied by the number of viruses that get out beta times the probability that each one of these viruses is able to infect a new cell, which depends on cell density. Right? So we have probability of lysis for washout, burst size, and probability of viral absorption. You could have done this cycle in the same way, focusing on virus particles, in which case it's focusing on a virus particle. What's the probability it finds a new cell? Probability it lyses times the burst size. And if that is greater than 1, then this can proliferate because one infected cell will lead to more than one, etc. And the same from the particle view. The consequence of this, of course, is that the invasion isn't just dependent on the features of the virus, but also on the susceptible population. Even with large burst sizes, if the susceptible population becomes too small, this virus will be unable to proliferate. And at a critical point, it will be. So the takeaway here is that ecological conditions with more susceptible cells and viral traits with more efficient interactions. Sorry, I just lost. Can you still hear me? Something happened on my screen that seemed to suggest my microphone is not working.
No, we can hear you.
Okay, fine. Good. Thank you. Sorry, I just went ahead and PowerPoint took over for a moment. Glad to hear it didn't affect you hearing me. So again, that more susceptible cells and viral traits with more efficient interactions favor a lytic antagonistic mode. And likewise, we can do the same thing with these latent viruses, building this model of a vertical infection. Here I have the probability of vertical transmission Q and horizontal transmission P, and I can focus only on the case where Q equals 1, ignoring the lytic pathway. And again, we can do these same calculations and ask what's the critical condition for transmission. It turns out our VER is simply equal to the time at which a cell is going to divide, which is 1 over D prime, that's cell death rate, multiplied by the division rate. But the division rate of these lysogens will be negatively affected by how many other cells are around because it has to compete with all these other cells. So we have division rate times cell lifespan. And if we then frame this in comparison, here's the positive relationship between the basic reproduction number for a horizontal strategy, and here's the basic reproduction number, 3 different options for a vertically transmitted strategy, noticing when there are benefits of lysogeny, it's higher, when there are less benefits, it gets lower, and here are fitness costs, but in all cases, it negatively depends on susceptible population because you have a rare lysogen competing with more and more other cells. And so that again suggests if there's reduced niche competition, direct cell benefits, or low variant survivorship, all those favor latent strategies. And just to point out, you can get intermediate cases. There are filamentous phage that bud rather than burst cells. And without belaboring the point, you can build similar models and end up getting these conditions for chronic invasion, which are an addition of the vertical and the horizontal transmission. And again, when this is greater than 1, you can find invasion. And the point here is that these intermediate strategies, and I consider temperate phage, which are using both at the same time, is also reflecting an intermediate strategy, suggests there can be opportunities for them to invade across a large range of susceptible populations, not only when susceptible populations are small. In this particular example, I've shown cases where this basic reproduction number actually has different domains in which one is higher, but of course the long-term fitness is not necessarily one-to-one related to the value of R naught. Only insofar as that is greater than 1, we at least know there's a chance for the invasion and evolution. And so that's really the takeaway point, that these temperate and chronic modes can be favored potentially along a much greater range. So finally, just to answer this question that I tried to pose, a step towards this, I think, requires a unified metric and requires thinking about virus and really phage bacterial interactions where we've predominantly thought of them, even in this obligate lytic mode, as predator and prey, to think of them, obviously they're obligate intracellular parasites, but as parasites, which require cells, and therefore thinking about their fitness, not in terms of the charismatic virion, but rather in terms of the number of infected cells. And in that metric, we have an apples to apples way of comparing lysis, latent strategies, and everything in between. And I'll try to wrap up here. I know I'm at 10:44. We started a few minutes after the hour, so I'll be sure to be done before 10:50 in the next 4 to 5 minutes. I just want to point out that in doing so, we have started to build much more complicated models that describe resource introduction, susceptible cells, exposed cells, a choice between lysis here and lysogeny there, and induction. And this gets complicated. Stewart and Levin had a model in which there was susceptible cells that got infected. If there was lysis, it was instantly, but you could have these lysogens as well. There might be a delay in some cases before the lysis happens. And Sylvain, working with Thomas Berngruber, also had similar models in which had fewer states. And so it gets to be a bit problematic to ask the question, okay, we have different kind of models. How much do we expect these to apply more generally? And these are not the only examples. There have been many different examples that take predator-prey viewpoints or parasite-host viewpoint. How much of viral fitness can transcend these model details? I'll just point out that in some new work that came out in Virus-Evolution a few months ago, we've calculated the basic reproduction monitor for all of these model classes and found, strangely enough, that when you calculate them, you get not just, we think of this as the average number of new infected cells in the next generation, but actually you have to go 2 generations forward. You get square roots because you have to think about not just 1, but 2 generations, because we have different life cycles or loops that have to connect over multiple generations. And happy to talk about the implications there, but we can end up using these diagrams in practice to do calculations. And just thinking about the R0 here as the contributions from single loops, and then because the double loops are squared, we have to discount them by a square root factor of 1/2. And I'll also point out you can get negatives, which often lead to people concluding it's complicated. We view these as a negative because these are disallowed loops. You can't jump from a lytic loop to a lysogenic loop. You have to pass through what we call a lysolytic loop, integrating, coming out, and then releasing again. And this gets to be a very busy slide, but I'll just to point out that in all of these models, the R0 calculation is literally this diagram. We have to weight them differently by the parameters and assumptions of the model, but we really have begun to use this kind of diagrammatic mode to do the calculations. Okay, and in doing so, you can see that in practice, these things turn out the same. That whether we're using implicit resources in which prophage benefits cellular growth or have a cost, as a function of susceptible cell populations, we can see that these horizontal strategies seem to do better as we increase cell densities. The vertical strategies tend to do worse, and intermediate strategies in some ways get the best of both worlds, avoiding positions in which R0 might actually drop less than 1 and be unable to invade. But this is just one step in the process. I mean, it's taken us a long time to get there, and I think the framework of thinking about infected cells is the right one to move ahead. In very nice work going back to 2013, Sylvain with Thomas Berngruber looked at an experimental system where they tried to evaluate, at least in the near term, the expectations for the relative frequency of a more virulent type to a non-virulent type, showing that when cells were available at the beginning, the virulent type increased in theory, but also in practice, and then was expected to diminish over time. Okay. And I'll point out that in addition to this interactions amongst 2 different variants, we also think that there could be ways in which this kind of, let's say, interactions between strategies might enable unexpected coexistence in the long term. Here's an example of an invasion of a temperate phage which is unsuccessful because it comes at a cost to the host. We then later introduce lytic viruses which increase and proliferate, driving down cell densities. And then we try in silico to reintroduce the temperate phage and find that they invade and coexist. And the idea is that the lytic phage have drawn down the niche competition so that these temperate phage, which don't provide a direct benefit, in fact have a cost, have a different benefit, which is they provide immunity to infection. And so that's a contextual benefit, which means that it's actually when lytic viruses are present, it opens up an opportunity for benefits of immunity to come into play, and just showing that this invasion basic reproductive number for the temperate phage, once there's a sufficient amount, this is no immunity to full immunity, once it passes a certain point, then it's able to invade, even though it has a direct fitness cost in terms of growth on its host. So to wrap up, I just want to revisit that original question I had. What is a virus? Whether it's a virion or a lysogen or a virocell, these lytically infected cells. And I hope I've at least convinced you to be curious about the idea that it's all of the above, and that we need to think more about the viral life cycle as being under selection. And viral fitness depends on measuring present and long-term value of infection across this life cycle, whether inside or outside hosts. And this won't just be a problem for experiments, but I think it increasingly will be relevant when we think about dynamics of virus and microbes out in marine systems, or even when we try to think about applying them in therapeutic contexts. So with that, I think I'll wrap up just to again say thanks to the many folks in the group and collaborators who've worked on this part of the story. We do work on other things, but in the interest of time, I tried to avoid them. We work on phage therapy with Laurent de Barbeau and Pasteur, and we're working on virus-microbe dynamics in ocean systems with many folks, both in the United States and abroad. And again, if you want to follow what we're up to, you can check out Twitter. Spend too much time on Twitter, and the code is released on our GitHub site, and we try to post everything on bioRxiv or archive, and sometimes these days medRxiv when it's COVID-19. Happy to stop and take questions.
Thank you very much. So I see no one asked questions in the Q&A tab, but we're gonna just try something new. And if anyone has a question now, they can just raise their hand. So if you click on the participants, you'll have the possibility to raise your hand. That worked well. Oliver.
I see one person gave me the French equivalent of the bird in the hand is worth two in the bush.
Yes.
I think Louis posted one in the chat. That was it.
I think he was giving me the
Not really. I was just replying by un tien vaut mieux que deux tu l'auras.
Right. That's precisely the sentiment of that expression.
So Ollie, you can ask your question.
Yeah, thank you, Joshua. That was a really, really nice talk. Thank you very much. I had one question that sort of goes back to older models of Mark Lipsitch, for example, where they show that the outcome or the relative advantage of going either horizontal or vertical depended a lot on the possibility of multiple infection. Because that determines essentially the availability of hosts for horizontal transmission. In your models, is that something you take into account?
So in the ones I've described, let me try to frame it. I totally agree, and I didn't have time to talk about some other work we're doing on this multiple infection problem. But you are right that, and I tried to allude to it at the beginning, that when multiple viruses infect a host, I think this implies host scarcity. In other words, and then if the virus kills that host, there may be very few cells, if none, to infect moving forward. Also with questions of superimmunity. In other work, we've extended these models, which do not include multiple infections, to include multiple infection, then ask the question, what should be the probability of lysogeny as a function of that, finding that it tends to increase with increasing multiple cellular infection. But I didn't go through those today. There's both this system and also a system from Rotem Sorik, and when there's a small peptide that seems to mediate whether or not these decisions are made. In separate work from about 2008, we then even tried to ask the question, because I've always been puzzled, even if that's the right thing to do, how do they do it? And so I didn't talk about that today. Our hypothesis has been that when multiple viruses get in a host, there's a switch. In the case of lambda, it's between, you could think of it as between the C1 and CRO pathways, and the duplication of genes you might think would not change outcome. But with nonlinear feedback, if you essentially have more copies of phage genes inside a target cell, because of feedbacks, you can actually shift the balance between these competing pathways in a genetic switch. So I think that in the long term, we'll have to reconcile the sort of intracellular, how to be temperate and have these plastic traits. But I agree with you that if we're to move ahead here, we have to explicitly include multiple infections in these eco-evolutionary frameworks.
Thank you.
I had a question. Well, actually, I have two questions, and they're kind of related to the same topic, which is trying to account for the dynamics of the system. What I mean by dynamics, the epidemiological dynamics. So the first one is about the theory, mainly regarding this quantity that you defined, the R0, and I think you clearly pointed out perhaps the limit of the approach by saying that it's only a first step. But what I wanted to highlight is that there is a very clear equivalent between this quantity and what is derived in other models of evolutionary epidemiology that account for the feedbacks. So there are ways to account for the fact that your derivation of the R node, there's always a S star, which is defines basically the state of the environment at the point at which you look at the invasion of the virus, and of course this environment is going to change as the virus spread, and it's going to feed back on the measure of fitness, and that's really complicated, and it's going to affect of course the dynamics and the invasion of different strategies. And but I know you're aware of that, but no, I just wanted to, given that feedback, I'm questioning when you use the R node, it's an epidemiological concept to discuss the invasibility, and I think what you have in mind is actually the evolution of the traits. So I think there is a tension between these two questions that you do not fully address with the R node.
So I think I'm going to agree with you, which is that, and I view this in part, our initial intention. Can you still hear me? I thought it went out for a second. That our initial intention here was to confront this predator-prey idea. And in fact, and I'm coming from a slightly different place. And so in fact, this is one of the things I wanted to do next year in France as the centerpiece of really trying to unify these ideas, which I think is a long-term goal. Within the environmental phage or microbial community, when you think about success, people tend to view it as really as the winner as being whichever phage makes the most particles. It's just, we count the particles. And so the first step here was, how do we even think about a strategy in a way that I think is, as I said, apples to apples, in a way that makes sense of what is under selection. And that is not the particle. The particle is not under selection. You can make a lot of wasted particles. If you don't get to host cells, it doesn't make sense. So that's just the first part. And I think in doing that, I have found that although we all know that phage are parasites, it actually in practice has not influenced the way that the field has approached, as you know, and in fact, like it's taken me a long time to adjust to that. These really are parasites, and we should take an epidemiology view, even though we think of them ecologically, right? And just that word is different. Now, that's what I was trying to bring in the last two slides, which is that once you establish an invasion, it implies many different kinds of things can happen. Mm-hmm. So when I think about the evolution of traits, then I have to think, well, we have certain values of temperate traits that could invade, but that is not an evolutionarily stable strategy. We have to think what's going to happen when those particular hyperparameters change. And then the reality and why this field has only gone so far is it just gets hard. It gets hard because they're much more diverse. They're usually not single pathogen systems. We can't wait until one reaches a stable equilibrium. So we can't make the sort of slow evolution assumption. We actually should be thinking in the fast evolution limit. And like you've shown in the near term, 20, 25 hours, it kind of makes sense, but beyond 25 hours, I'm not sure we know what's going to happen. And so I tried to allude to the end that this is a step in which I think it's giving us a principle to understand a sequence of invasions, like you did with Thomas, what I was trying to allude to in these chronic examples. But in terms of long-term strategies, I don't know the answer. That's something that I think we all need to think more about. I'm hoping that the infected cell paradigm can help us make sense of it as the right way to frame our metric for a Malthusian fitness and ask that question appropriately in the long term.
Right. So
it gets a bit philosophical, but I think you were asking a philosophical question in some sense. So.
It is, but I think there are some tools that can be used. Like, so you're talking about class structured models, and on top of that, there is temporal dynamics, and there are tools that can be. But I have a second question, if I may, is about the empirical data that you discussed, and I think it's related also with this philosophical question, maybe. Which is, so you're trying to explain, or others also have been trying to explain the pattern that you see in the ocean, and I think you were very right in the different papers that you wrote about that. That we need mechanistic models that are able to produce those patterns, but I was wondering about the first result that you showed was about seasonality. And I was wondering whether people account for that seasonality, because when you measure the ratio of virus to microbes, don't you think that it would be important also to account for the temporal dynamics through the year, where, I mean, this ratio is, if there is seasonality, this ratio is doomed to fluctuate as well. So I wonder how, I mean, maybe this dimension through time, through the year, could also help explain the diversity of the patterns that you see in nature. And I was wondering whether people have tried to account for that.
Yeah, so the brief answer to that is that often these studies go out in a certain period of time, usually in the summer, wherever the summer is, because being in the winter in the ocean is not so much fun. So they're often in focal periods of time. We have in other work, which I didn't talk about today, have these sort of temporal dynamics, and these are often focused, we've started largely on obligately lytic cyanophage systems because we can get relatively high resolution data. There's a paper that just came out in ISME journal by Merwat et al. With Debbie Lindell as the senior author, and we have models now we're working on that try to actually fit time series data increasingly over multiple diel cycles is typically what we're doing rather than seasonal cycles to start. And I can say that I'm relatively confident that at least in these surface ocean systems, we can explain a lot with these simple models. What becomes complicated is that not only are there differences between seasons, but also the structure. There can be more particles, and particle-associated dynamics are very different than the planktonic-associated dynamics. And we think there may be more lysogeny going on there and other kinds of interactions in those domains. So I think there are many ways to critique the data. I could have critiqued it also by diversity, that there's a whole diversity of systems, and now we're going to add them up together and expect the pattern to be replicated by a one-dimensional system. In fact, that to me is the most direct approach. But nonetheless, our point in those papers was just to show that sometimes we're asking too much of patterns. And I think we need more work that actually links the pairs together. Because that's the advantage of the experimental approach, but we go to these natural systems when, in addition to having 10 billion viruses in a liter, it's not 10 billion of the same type, right? These are incredibly diverse systems. And so the more we connect who's infecting whom, we'll have a better chance of actually understanding mechanisms as opposed to trying to infer them from in some sense, an integrated signal that washes out a lot of the info. But yeah, so I didn't get time to talk about the time series modeling work, but some of it should be available already on bioRxiv, ISME-J, and some will come out soon.
So Nacho, you have a question.
Yes, great. Thanks a lot. I mean, I have liked very much then your idea of not thinking of viruses only as the virions themselves, which is something that we usually measure and then we conceive them just as but actually, so this viral lifestyle, so how this evolution, this diversity of viral lifestyles is. But then I would like then also to ask you a question, I turn onto this, which is what is a host, and then what do you think of what a host is? And then I personally work on viruses that infect multicellular organisms. So, and then I would like them to hear your opinion on whether the host is the multicellular organism, the individual cell that is eventually killed or not, and then how then should we then conceive fitness here, in the sense of it is the fitness of the cell that is eventually killed, or is it the fitness of the organism that may not care at all of one of his or her cells being killed? What do you think of what is a host then?
Do I have to accept one of your two options, or can I say both? Or
you can take any or both or none of them. I mean, no.
Okay, so I mean, I certainly think, I'll give you just one example of why I think you're asking the right kind of question, why it's going to be complicated. So one of our collaborators, Alison Buchan, has looked at a very interesting Rosiobacter system in which the Rosiobacter released these phage that occasionally killed the Rosiobacter, but with a competitor seemed to knock out the competitor and vice versa. So you then have an instance more of a coalition, like what is the virus and what is the host, because they seem to depend on each other almost on a mutualism, but are antagonistic in other contexts. So once you have the integration event, then this incentive structure between versus defense versus letting in, the benefits of the viral infection can be significant. And the more that the virus sticks around with the host, the more that it spends time vertically transmitted, the more that's true. Though, of course, there's a risk for the virus because all of the genes associated with horizontal transmission will, for the most part, be under neutral selection and then eventually become defunct, right? So I guess I would just answer the question by saying that I certainly think that the viral fitness we should view in terms of a life cycle, but because they're spending time with the host, these are entangled in a different way that I didn't elaborate on today, and I can't claim to totally understand it, but certainly the host fitness will also depend on its parasite load because these parasites are often helping the host, right? There is a kind of a holobiont here in a coevolutionary sense. So I'll leave it at that. I don't think I know more than you about how to describe that yet, other than that it is entangled.
Thank you very much.
Joshua, I don't know about your time schedule, but would you mind taking another question?
Sure.
So Giacomo, if you want to ask your question, you can go.
I realize it's late in the day in France, it's 5, and on this end, we have some COVID-19 matters to respond to mid-morning. Otherwise, I would be more willing to stick around for longer, but I'm happy to take another question.
Okay, one last one.
Yeah, hi. You talk about the switch between basically horizontal and vertical. So I was wondering whether these phages can, or what do you think about these phages losing the switch, and if you know, or the selective pressure moving to just one or the other cycles, and if it's evolutionarily possible for these phages to do it.
Yeah, the switch from having the ability to do both things to only having the ability to do one is possible, and these are known lambda-vir mutants that if you delete the appropriate genes, and this can obviously happen, you delete the ability, you remove the ability for the virus to be temperate and become obligately lytic. Going the other way, I would say, is a much harder process. So I would say that in terms of strategy sets, thinking about parameters as being evolvable, meaning the probability of initiating lysogeny, which I want to point out is a plastic trait. It is not a fixed number, although we often assume it's a fixed number in some of our work. Ultimately, we should think of this as being dependent on co-infection numbers and host state and other features, and same with the induction probability. There can be a spontaneous induction rate, but there's also going to be a sensitivity to stress. There's a way in which, for example, phage lambda can listen in to the SOS system, but phage may also be able to listen in on cell density by essentially having their gene regulation tied to quorum sensing genes and other ways in which cells are communicating with each other. So my brief answer is yes, there are ways to move from temperate to lytic. The other way is not an evolutionary easy path. It's not to say it can't happen over some very long period of time with gene transfer, but one way is quite direct. And the evolution of traits within the temperate or the virulent space is certainly possible.
Thank you.
Yeah.
So then, well, thank you very much for giving this really interesting talk, and thank you, Sylvain, for suggesting Joshua as a speaker. We are going to end our day here, and I hope you have a very good Friday. I hope everyone has a nice weekend, and we'll be back next Friday at 11:30. Thank you very much.
Thanks so much. Thank you.
Bye.
Bye-bye.